Hello, everyone. Welcome to today's live broadcast of the topic, Optimizing the Conduct and Implementation of Animal Studies. Today's presentation is titled, The Future of Preclinical Animal Models in Pharmaceutical Discovery and Development. The scientific 3 R's of relevance, robustness, and reproducibility are critical. My name is Julianne Chaya, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. We have a few important announcements to make before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right side of the slide window. If you have any technical problems viewing or hearing the presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speaker, Dr. Jeff Everitt. Jeff Everitt currently serves as the Director of Comparative Biology and Medicine at GlaxoSmithKline R&D in Research Triangle Park, North Carolina, and also serves as the Adjunct Professor in the Department of Pathology at Duke University School of Medicine. He received his doctorate in veterinary medicine from Cornell University and then conducted residency and postdoctoral training at the University of Pennsylvania and the University of Illinois. He is a member of the American College of Laboratory Animal Medicine and the American College of Veterinary Pathologists, and has served in leadership positions in both organizations. He has led animal care and use programs and has been an active advocate for increasing the quality of animal care and use and the translational relevance of animal models. He is a former council member of the Institute for Laboratory Animal Research of the U.S. National Academies. I will now turn it over to Dr. Everitt for his presentation.
Thank you, Julianne. Welcome to everybody, and what I'm going to talk about today is something I want to introduce, which is really the topic of this whole virtual conference, which has to do with what I'm calling the scientific 3 R's of relevance, robustness, and reproducibility. And the objectives of my talk today are to review some of the present challenges that are being posed to the use of animal models in drug discovery, and to discuss the framework of how animal model research can be improved and the potential value of laboratory animal scientists to this process. I want to introduce these scientific 3 R's, which has really already been introduced in this conference for those of you that have attended other talks, and review some of the trends in the external research community that I think are going to significantly impact animal work in the future. And really, this talk and this whole conference is to provide a call to arms for laboratory animal scientists to rally around and to improve the conduct of in vivo studies. Now, why is this necessary? And one of the things that struck me was a recent editorial in the British Medical Journal which brought up the question of how predictive and productive is animal research. And it's one thing to hear this from the animal welfare community, but it's another thing to see this in a major medical journal. And then there have been a number of prominent publications. This is one from Nature, which calls into question the quality of animal experimentation. And here you can see just in the title, quite provocative, animal experiments under fire for poor design. And there's been an awful lot of criticism that's been thrust upon the animal using community of late that really doesn't have to do with animal welfare, but has to do more with what I would call experiment welfare. And there's a number of common threads in this critique. One is lack of experimental rigor, failure to remove bias, failure to report properly in sufficient detail, study design errors, and questioning the biological relevance for the questions at hand. And I think that these questions should be extremely important to us, not just for those of us that work in the pharmaceutical industry trying to come up with disease-relevant interventions, but really for the entire animal-using community. And one of the things that I've been very interested in as an animal model support scientist in the pharmaceutical industry is the translational value of models. And here you can see that over the past 2 years there have been a number of prominent publications that have really called into question the translational value of some of the animal studies, and these were picked up in the popular press. And certainly the title on the top got picked up by the New York Times, and it's really created quite a stir. And I think that increasingly in the future, attacks on the use of animals in research is going to be based upon the science rather than some of the criteria that have been used in the past, which have been primarily welfare-centric. In addition to the questions about translational value, there's been a very serious questioning of the quality of the research, and there are quite a number of talks in this conference that I would refer you to, having to do with reporting of studies and designing of studies, and the entire animal using research process has been suffering at various in its reproducibility for issues of what I call quality. And we're all of us that are laboratory animal scientists are very interested in the quality of animal studies, and it's going to be important to really get this in hand and take a look at this and why both in the popular press and in the scientific press now there has been a serious questioning of the quality of some animal studies, including very prominent studies. Now, the way I like to look at animal care and use, and this transcends the issue in the pharmaceutical industry, but is really the use of animal models in general, is that there has to be a balance between welfare for the animal and welfare for the science of the experiment, the question at hand. And if you see on the right there, you can see the NIH poster that's been around for years that says good animal care and good science go hand in hand. And of course, we all know that to be true. But animal welfare and experiment welfare are not one and the same. And at times, I personally feel that laboratory animal science has been driven by the animal welfare agenda a little bit out of balance and at the cost of the scientific agenda. And I think it's important that we bring this back into balance. And I think that addressing these scientific 3 R's and putting them at the same prominence that we've had in the Russell and Birch 3 R's of reduction, refinement, and replacement, I think would go a long way to address that balance. So what's the crux of the issue? The crux of the issue is that we need to better plan our animal use relative to very focused questions that we're trying to address with animal studies. We need to apply better experimental design to many of our in vivo studies. There's a need to conduct studies in a manner that minimizes bias, and some of the earlier talks today, those of Malcolm McLeod, for instance, I would definitely recommend the audience listen to if you haven't already heard them. And there's really some excellent work that's been put forth in this particular conference describing why we need to minimize bias and various ways in which it's best to do that. And the reporting of research needs more rigor and transparency. And there were also talks, and there are going to be more talks on guidance for publication, and there were talks this afternoon about, from the standpoint of journal editors, of what is important in the reporting of research. But I think most importantly, it's important for all of us to realize that laboratory animal scientists have a key role to play in this enterprise-wide shift to increase the quality of in vivo studies. Now, in the current approaches to drug development, there has been recently tremendous shifting of how animals are used in preclinical drug discovery and preclinical drug safety. So in other words, there's been a huge increase in the number of non-animal platforms in high-throughput approaches, in very, very powerful omic approaches to really dissect out pathways and really get a lot more translational science underpinning how our pharmaceutical compounds are not just discovered, but also how they work. And despite all that, animal studies are still quite pivotal in decision making, both in pharmaceutical discovery and in pharmaceutical development in many therapeutic areas. So it is important to the pharmaceutical industry, as well as in animal research in general, to really get a handle on increasing and maximizing and optimizing how we conduct those animal studies. And it goes beyond just deciding, well, what's the proper model? It really comes down to really focusing what's the proper question to ask, and then what is it we're trying to do to answer that question before we can even get into the issues of the model. And then when deciding on the model, there are issues of translational value of the model, and there's also quality issues of how the model is conducted. And so all of those things go into a holistic assessment of how an in vivo study is put together. Now, in the pharmaceutical industry, there's been a tremendous change in the landscape of animal use. There's been a big impact of non-invasive imaging technologies. There's a lot more telemetric monitoring going on. We're automating many types of sampling. In general, animal use has been going down, but the animal models are getting complex, and there's a lot more multiplexing of endpoints. There's also going to be, I think, a renaissance in the use of genetically modified animal models to an even greater extent than exists now with the advent of new technologies like CRISPR and TALENs. So this makes it, in my view, even more important that there be some concentration now on what I'm calling the scientific 3 R's, and that's to make sure that animal studies are relevant to the question that's trying to be addressed, that they're conducted with robust kinds of assays, and that the studies are reproducible, and not just that they can be reproduced, but we really do need to reproduce them. And it is, I think, been a little bit of a drain in some areas that animal research has become so expensive, and the time and cost constraints are so much in animal-based research that many studies really are not reproduced because there's a pressure not to reproduce them, to minimize animal use in some cases. And I think it is important for the question of how robust an assay is and how reproducible an assay is for multiple laboratories and animal models to be performed more than once with many of our important studies. These scientific 3 R's are not intended to replace the 3 R's of Russell and Birch, but rather to place emphasis on the health and well-being of the experiment rather than just the health and well-being of the animals in the experiment. And in the pharmaceutical industry, and I think this can be generalized in a lot of mechanistic discovery work in academic centers as well, there is a big push now to really make sure that we understand the translational quality. The pharmaceutical industry is trying to reduce the attrition of compounds through the pipeline, and it's concentrating on really trying to increase the quality of targets that are being selected, as well as the quality and druggability of the molecules that are used to impact those targets. And all of that pertains quite extensively with the translational quality that allows the preclinical program to predict and have relevance for the clinical program that's selected. And this is something that every company and every drug discovery team is certainly enmeshed in right now. And one of the reasons for that is that the contemporary paradigm of drug development is really an unsustainable model. The cost is just skyrocketing. It currently costs over $1 billion to bring a drug to market, and that's an unsustainable cost. And so the industry as a whole is trying to reduce its late-stage attrition rates. And in order to reduce those late-stage attrition rates, it's important for the preclinical program, both the non-animal and the animal portion, to be predictive of what's going to happen in the clinic to the extent that it's possible to make that prediction. And so every company has strong emphasis on trying to reduce the attrition of drug development. So trends in the pharmaceutical industry in vivo and non-clinical efficacy research are that there's a diminution in the overall animal usage, particularly in discovery areas. There's a diminution in the use of non-human primates as well as other non-rodent models. There's been an increased complexity of the models and multiplexing of readouts. There's a very, very significant increased emphasis on the use of human disease-relevant phenotypic screens and increased externalization of discovery activities to partnering groups, either contract research organizations, academic centers, or public-private partnerships. And there's also a lot of increased pre-competitive and collaborative activities across the pharmaceutical and biotechnological industry. One of the challenges that we have, and several of the earlier talks alluded to it, is that it's very, very difficult to depend on the published literature for validity of research. Many of the targets that are selected for drug development come from the literature, and many of the animal models that are chosen for trying to discern different molecules in lead optimization programs are based upon animal models that are published in the literature, and there's many, many biases that lead the literature to not be something that can be easily trusted. And much of this work now has to be reproduced internally within the laboratories of companies. And preclinical irreproducibility across the board is a systemic problem that's driven by the current research incentives that are out there in the publish-or-perish academic environment. Careers are built on publications in top-tier journals. Top journals want simple, compelling stories. It's easier to publish positive studies than negative studies. There's little recognition when studies can't be reproduced, and there's little encouragement for publication of confirmatory data. So the literature is not safe for the pharmaceutical industry to just delve into and take on for next steps. And much work has to be reproduced. And many people are probably familiar with publications that have come out of Amgen that were published by Glen Begley, and he was in a hematology oncology group at Amgen, and they looked at 53 targets that were published in top-tier journals and tried to reproduce the work in the laboratories at Amgen, and found that a very high percentage of the published work could not be reproduced. They then went back to the original laboratories that produced this work and tried to work with those laboratories to get them to reproduce their own work, and there was difficulty in getting the work to be reproduced. And this really called into question the quality of published targets out there in the literature in the oncology area. And there were a number of common faults in the reporting of much of those experiments, including whether the studies were blinded, whether all the results were shown, whether there was replication to the experiments, were the controls proper, did they show positive and negative controls where appropriate, validation of reagents were often a problem, and often the statistical methods were inappropriate. So there were a lot of common problems that arose when the literature was reviewed, which really brought up the issue of, do we optimally plan and execute animal studies? Now, there are some papers that have recently come out of the National Center for the 3 Rs in the UK that underpinned the ARRIVE guidelines, which are going to be discussed in detail at this particular conference. But it was really a survey of a variety of reviewed publications across biomedical research, and what Carol Kilkenny and colleagues found that there were numerous common deficiencies across 271 publications that were reviewed across a whole variety of journals. And they found that Only 59% of the papers that they reviewed even described objectives and hypothesis adequately. Only 70% of those papers described statistics. The vast majority of the papers didn't discuss issues relevant to preventing bias, such as blinding and randomization, and features that we know are extremely important in the output of in vivo studies. And so that led to the development of the so-called ARRIVE guidelines for reporting animal-based research. And really these guidelines are a checklist of 20 different features that are believed important in the description of studies. And what I would say is that many institutions are adopting, if not the ARRIVE guidelines themselves, then at least the spirit of the ARRIVE guidelines by looking at developing criteria for the description of in vivo studies. Malcolm McLeod, who spoke earlier in this conference, has been a pioneer in sort of pushing for having good in vivo laboratory practices. This is from one of his older papers, but talking about the importance of sample size calculations, inclusion and exclusion criteria, randomization, allocation, group concealment, and how animals are to be excluded from analysis and how they're to be reported. And these all are important things for in vivo scientists to consider when not just designing but also conducting in vivo studies. And I would say that the devil's in the details, and it's very, very important to consider the type of study and what the potential readouts from the study are. It's hard to just come up with a single checklist that takes care of everything in all studies. But it is important for every individual that's conducting in vivo research to, in the design of their studies, to consider the ARRIVE guidelines, to consider these various checklists that have been formulated by different journals that they publish in, and to design their studies with the end in sight. And so that's something I think is very, very important for all laboratory animal scientists to consider. It's also important to consider that experimental problems come at all stages of the life cycle of an in vivo experiment. So while much has been said at this conference about deficiencies in the design of studies, there are also a lot of issues that laboratory animal scientists are most frequently faced with in the execution of studies. And then obviously in the analysis of studies and in the interpretation and reporting of studies, there are important things to consider. I think that for laboratory animal scientists, though, it's very, very important to consider the details of the execution of a study. And it's very, very hard to publish all of the details of the environment of an in vivo study. Everybody's aware that you can't put your entire ALAC site visit document in the materials and methods of a paper, but there are many, many things in that programmatic description that could impact an animal study. But it is important for every investigator to consider what are those things that are most impactful to creating bias and to creating variation and possible implications to the replication of their studies, and to make sure that they address it in all the stages from design and conduct right through analysis and interpretation. Now, much has been said today about two of those practices that are used for increasing the quality of preventing bias, and those are randomization and blinding. But I would say it's not a simple one should randomize and one should blind. I think that the fallback position should be randomization and blinding, but it's certainly not necessarily appropriate in all experiments, and also it's important to describe very carefully how randomization is done. And randomization can be done in a variety of different ways and randomized according to a variety of different criteria depending on what the nature of the experiment is. It's also important to consider the readouts of an experiment and where problems occur in vivo experiments, because not all the problems of an in vivo study fall into inadequate description of the animal. It can fall into inadequate validation and description of the assay that the animal is being used in that in vivo experiment. And so how in vivo assays are developed and how the replication strategies are designed are extremely important to consider. And that documentation of the development process is absolutely critical, and the replication strategy. Now, this is not a new issue. So this issue didn't start with the ARRIVE guidelines, and in fact, there's been advocacy of checklists since 1985. And so it's been well over 25 years, and I think that we really need to start asking the question of, well, why haven't these things been adopted? And there's a variety of reasons that the checklists haven't been adopted, and I'm a little bit skeptical about all the checklists that are out there. Checklists are very important. Checklists are very useful tools, but we have descriptions of gold standard checklists, and the next one will be the platinum standard, and then we'll have titanium standards, and every time somebody comes up with another unknown variable on an experiment and tries to control for it, we'll have another checklist. Checklists are just a tool. What's really important is to go through an experiment with the thought process of what do we know? And that's where earlier talks on systematic review and how to properly review what's already been done is important for discerning where things can go wrong. And it's also important to consider to connect principal investigators and experimental design with those individuals that actually conduct experiments, because they generally know where the critical phases are and where studies can go wrong and where bias is likely to be generated in an experiment. So it's very, very important to consider the life cycle from design right through analysis of results of where bias is going to come in or where bias may have crept in, so that it can be addressed and properly reported with transparency. And as an example for the customization of checklists, many of us are now using pretty sophisticated non-invasive imaging modalities, and in imaging papers there are many animal-related issues that need to be very carefully described, and those same issues don't necessarily need to be described in, for instance, a surgery paper that doesn't use imaging. So there are things that need to be described in every paper, and then there are very important details that need to be described in certain kinds of papers and certain kinds of studies. And I think that it's extremely important for journal editors to look at the ARRIVE guidelines, look at existing guidance that's out there, and then for their journals customize those things that they think are most important to properly describe in detail. Because the devil is in the detail when it comes to describing in vivo studies and in conducting in vivo studies. This is from a recent paper that I was involved with, which is looking at what's the minimum information to describe on the pathology endpoints on an animal study. So the endpoints can affect a study. Many times animals are only used to generate diseased tissues that are going to be studied, and often the lack of reproducibility or the variation in studies are due to issues in tissue handling rather than animal handling. So it's necessary to tie together the readouts on studies with the actual conduct of the animal study itself, and it has to be looked at in a holistic framework. So in the review of animal studies, I think it is important to look at the review process really in two different ways. One is in the ethical review process, and in the United States, really what that constitutes is the Institutional Animal Care and Use Committee process, and that really, to me, deals with the traditional 3 R's. Can you replace your proposed method with a non-animal method? Can you reduce the numbers of animals you use? Or I would rather than say reduce the animal numbers, I would say, can you optimize the animal numbers through optimal design? In some cases, that's use more animals, but use optimal design. In other cases, I think refining animal methods, and certainly IACUCs deal with refinement by assuring the minimization of pain and distress on experiments, optimizing animal welfare during the course of that experiment, and doing a variety of animal-related refinements. That, to me, is one type of review. It's critically important. It's necessary. It's codified in many kinds of animal-using regulations all around the world in various forms. In the U.S., it's in the form of an IACUC. In Europe, it may be in the form of ethical review committees. But I think it's important now to go beyond IACUC reviews with really peer interactions and peer reviews on the science of in vivo experiments. And that goes beyond the things that most IACUCs generally consider. And that really looks at what I'm calling the three scientific R's, those of the relevance of the model, the translational relevance, the questioning of design with respect to biological plausibility for the questions being asked, the robustness of the in vivo assays and what's likely to impact them, and the reproducibility and replication ability of those assays. And that, I think, takes a different kind of expertise. It takes different kind of peer interactions and different type of review than the ethical review process. And I think to optimize the use of preclinical models, not just in the pharmaceutical industry, but really throughout in vivo studies, I think it's important to have both of these kinds of committee and peer-related interactions. And we all know as laboratory animal scientists that there are many different things that influence data. Everybody's heard about all the changes now that are coming to light with studies of the microbiome. Microbial status is important as far as pathogens, diseases and background lesions of animals, the genetics of models, the environment that these animals live in is critically important. Study methods themselves influence data to a tremendous degree, as does the animal care and use program. And there are a number of tracks that at this conference that deal with the training of people and what in the program influences data. But there are many, many things in that program that influence in vivo experiments. And there are many sources of generic variability in studies. In the pharmaceutical industry, we have a lot of questions around compound and formulations, let alone dosage and dosage administration, animal issues, environment issues, assay types and equipment. There's a lot of operator issues. Replication and robustness may depend on how many individuals are involved in the assay. And then there are many, many issues that affect variability when it comes to the analysis and reporting of results. Now, with that all said, I'm going to talk a little bit about how in my work at GSK, how we look a little bit at some of the issues of translational science with preclinical models. And I want to credit a number of my colleagues at GSK, primarily Brian Berridge and others in discovery pathology, with coming up with an animal model selection assessment framework that we use for translational relevance. One of the first things we do is we ask what the human condition is that we're trying to develop interventional therapies for, and what is the disease and the disease entity that we're trying to develop that therapy for, and how well do we understand it. Okay, so in order to model something, you have to understand what you're trying to model, and you have to understand how the model that you're choosing to use is going to model the process. And so we ask how well characterized the etiology and pathogenesis of our patient disease population is, how well characterized is the role of our putative molecular target in the intended pharmacology, how discrete is the line of sight from our intended pharmacology to the clinical outcome of interest, and how well is our intended patient population characterized with respect to demographics, genetic heterogeneity, comorbidities, and other issues that affect the disease process. And also what's critically important is understanding if there's a characterized and translatable biomarker that would be available to help bridge between the preclinical program and the clinical program. We also are interested in aspects of preclinical model physiology. How well is the systemic and molecular physiology of our intended patient replicated in the model that we're intending to use? Does that model replicate the relevant systemic anatomy and physiology? Does it replicate the relevant molecular physiology? This is critically important, particularly as our therapies get more and more specific around very specific intended targets. And often we're trying to modulate very, very exquisitely sensitive and specific pathways. We want to know about how the model pharmacology stacks up. Does the model replicate the expected pharmacology in patients? Is the molecular target conserved in the species that we're trying to model in? Is there evidence that modulation of the target in that species produces a relevant biological effect? Is there evidence that the modulation of the target is going to have a similar effect as in patients? So it's also, that's just the primary pharmacology. It's also important to know about possible secondary pharmacologies that are affected when you hit the target. And we want to know whether the model's positioned to characterize that secondary pharmacology, because that can have very, very important unintended consequences. And we want to know whether that translatable biomarker of pharmacology exists so that we can go between our preclinical model with clinical patients. We also want to look at how well our model may model disease. We're often, as veterinarians, most familiar with disease models, but really disease models are only one subset of the total preclinical modeling picture we're using. And in many cases, we're not trying to model a disease in an animal that's present in a patient population. We all know that there are profound differences between animal diseases and human diseases. But in the case of disease models, we want to know if the etiology is the same, is the pathogenetic time course similar, how well does the model replicate the clinical outcome, does the model replicate the clinical phenotype, or is the phenotype different? What about the pathophysiologic pathways that lead to the phenotype? Are they similar? And does our intended study design replicate our intended clinical trial with the dosing regimen that's going to be used to affect the patient population? It's also important to know whether there are disease comorbidities, and are we replicating those comorbidities in a relevant way in our animal model. And also here in disease models, it's important to have translatable biomarkers when and if possible. So using those four categories of looking at our animal model relevant to disease patient populations, that makes up what we call our translational matrix. And those go to the 1R of relevance. And that's how we look at, is the model we're choosing relevant to some of the patient questions that we intend to address. And it's very much that framework that helps us bridge the preclinical program in the discovery areas to ultimately the intended clinical outcome. Because one of the things that we really are trying to do in trying to reduce attrition in the pharmaceutical industry is make sure that the thing that's paramount, obviously, is patient safety, but we also do not want compounds to fail late stage for lack of efficacy. And really to show that something is efficacious, you really have to consider all of these questions in that translational matrix. So, really in starting to summarize this, I would say there are a variety of opportunities for improvement of how we conduct animal models. I think that we have to deal with both the translational relevance of our models, but also there are tremendous opportunities to improve the experimental design, the experimental conduct, as well as the experimental reporting, analysis and reporting of in vivo studies. And so I think there are profound quality issues that we as the lab animal community are going to have to address, and there's also animal modeling issues that I think we as an in vivo scientific community are going to have to really get our hands around in a much stronger way in the future. I think there's improvement in the quality of many of the things we do with our animals. Not just, I think over the last several decades we've certainly increased the quality of our animals, but the use of our animals as reagents still can be improved. We're learning more all the time. I think the microbiome emphasis of the last 2 years is really going to translate into us looking much closer at our animal models with even more rigor. I think in general that in vivo studies really have to concentrate on the reagents that are used. In the pharmaceutical industry, there are issues of formulation that are absolutely critical. It's one thing to properly dose an animal. It's another thing to properly handle the compound and the excipient in the proper way, in blinded fashions, with group concealment, not know what you're dosing. It's very few animal studies are actually double-blinded. And many of us at times feel that if we conducted clinical trials in the manner that we conduct our preclinical animal studies, with very small groups of, 6 to 10 people, and we did it in a non-randomized, non-blinded fashion, we would be quick to say that people don't translate to other people. And certainly there have been many late-stage problems in clinical experiments where the early stages of putting formulations into people didn't seem to predict. So it's critically important to make sure that there's rigor at all steps of this process. And I think there's a lot of room in the reporting of studies for journals, review bodies, granting bodies, and scientific oversight bodies to make sure that there's less bias in the system in selecting positive rather than all studies that have been conducted. And we all have to make sure that we don't demonstrate behaviors of failure to reject our favorite hypothesis. And I would say that this whole area has been very important, and that's why the committee at this conference chose this particular scientific topic as the lab animal science topic this year, but it also was the topic of an ILAR roundtable at the National Academies where reproducibility of animal-based research was the focus of a workshop that brought together many different stakeholders. And it was very clear at this workshop that this quality issue in the conduct of in vivo studies really has to be looked at with more rigor, and the reporting of studies has to be revolutionized really in how journal editors deal with these things. And there have been a number of important topics raised at this conference that I think will be the subject of laboratory animal science over the next decade, to be honest. And one of the things that I think is important from the standpoint of those of you that are in preclinical work in the pharmaceutical industry is the need to really distinguish between exploratory and confirmatory preclinical studies. So I think many academic and many mechanistic studies are what I would call exploratory studies, as well as many, many industry studies. But there are times where, and I think exploratory studies, while they require the same design, conduct, analysis, and reporting rigor as do confirmatory studies, I think they're of a different nature than confirmatory studies, and I think that you can get away with a lot more. In confirmatory studies that are really mini challenge studies or mini efficacy studies, looking at will a preclinical model serve to demonstrate efficacy of a therapeutic intervention, these have to be conducted in a much more rigorous fashion, and I think in a much more analogous fashion to what we've learned in the clinic with the conduct of clinical trials. And there was a nice earlier discussion today by the group in the Netherlands looking at the use of systematic review as an aid in designing studies, and also mentioning the Cochrane collaboration, which has really been at the foundation of increasing rigor and quality in clinical trials. And for the first time now, they're taking up the issue of how preclinical animal studies are conducted. And I do think that there are very strong linkages to make in how we do our preclinical efficacy work, in then bringing the results of that work into the drug development paradigm for taking into the clinic. And recently, there's been some discussion of multi-center preclinical randomized controlled trials looking in preclinical models at therapeutic interventions, putting the data together with well-construed meta-analysis, and really conducting those preclinical studies across academic centers in a way that's very analogous to how work's conducted in the clinic. And I think that we're going to see more and more of this in the future. So with that, I'm going to conclude by saying that I really think that this conference ought to serve as a call to arms, that steps need to be taken to improve the planning, the conduct, the reporting, and analysis of animal studies. I think the laboratory animal scientists who work in the discovery support space have a very significant role to play for implementing what I would call the scientific 3 R's of relevance, robustness, and reproducibility. And I'd like to leave everybody with the thought that the health and welfare of the experiment is just as important, and perhaps more important in many respects, than is just looking at the health and welfare of individual animals on those experiments. And with that, I'm going to stop, and we can take any questions. Julianne, are you there?
Yes. Hi. Please, so thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will try to answer as many questions as we can. I'm now just waiting for some questions. Thank you. Great, we have one question from Oliver.
So there's a question on how diseases in laboratory animals are modeled for ethnic variability in diseases, and that's an extremely good question. There's a big effort in the pharmaceutical industry to really to try to understand in patient populations the genetic underpinnings of disease. And certainly the polymorphisms that are present in the patient population are studied by genetics groups within the pharmaceutical industry. And where appropriate, obviously species, strain, and stock differences are taken into account for trying to understand how those polymorphisms may translate into research animal populations. And there are also many new technologies that are coming out in genetic animal models. Many of you are probably aware of things like the collaborative cross in the mouse biology arena, and there are a lot of new and potentially important ways in which genetic modification of animals are going to be used in the future for trying to model those differences in the human population and into trying to aid us in the development of personalized medicine.
Okay. If there are no more questions, I would like to once again thank Dr. Jeff Everitt for his presentation. Do you have any final comments?
No, other than to encourage the audience to attend many of the other talks in this conference. I think that the subject matter of this conference was summarized a bit in my talk, but each of those various areas of the scientific 3 R's is, I think, critically important for us to continue to work on, and is the subject of more detailed discussion in some of the other presentations. So thank you to everybody who attended.
Great. Today's webcast will be
Julianne.
available for on-demand viewing through 6 months from the live date. You'll receive an email from Roberts alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.